at KSR Siddhartha College of Pharmaceutical Sciences in association with Indian Pharmaceutical Association Siddhartha Nagar branch. Now it's my privilege to invite Dr. T.P. Rao, sir, Associate Professor, Department of Pharmaceutics and Biotechnology, whose expertise has significantly contributed to the academic excellence of our department. Dr. T.P. Rao, sir, I request you to kindly take over the virtual podium, moderate today's afternoon session, and steer today's proceedings. Thank you, sir. Please begin the session.
Hello, good afternoon, everyone who is attending this session. And I would like to introduce Dr. Nilesh Kumar Mehra, and he is a distinguished pharmaceutical scientist and assistant professor in the Department of Pharmaceutics at the Naipur, Hyderabad. With over 13 years of academic and experience, he has made significant contribution in the field of drug delivery and pharmaceutical technology. Sir has earned PhD Dr. H.S. Gaud postdoc from Irma Lerma Rangel College of Pharmacy, Texas, A&M Health Science Center, Kingsville, Texas, USA. He served as manager in product development, Scientist Research Center, Scientist Pharma Private Limited, Gurgaon, for development, scale-up, and technology transfer of complex ophthalmic inhalation and optic pharmaceutical products. He received Team Award for successful commercialization of ophthalmic suspensions product. He also received the Shakuntala Amir Chand Award in the Clinical Sciences from Indian Council of Medical Research, New Delhi. Dr. Nilesh sir has authored more than 130 peer-reviewed publications in high-reputed international journals and more than 50 international book chapter contributions. Dr. Mehra has H-index 44 and I-10 index 88 and total cumulative impact factor around 600 and over 5,500 citations reflecting the global relevance and impact of his research. He has 4 granted and 4 filed patents on manufacturing process and composition to improve therapeutic efficacy for topical delivery and 1 successful transfer of pharmaceutical technology to industry. He has completed 3 academic research projects and 2 industry-sponsored projects. Sir has participated in academic and scientific collaborations across Europe and the USA. He has supervised 5 PhD students and 10 PhD students are currently working. He has guided 36 master students for their dissertations, research projects. He has received numerous outstanding awards, including Young Scientist and Team Awards for their research output. He is the recipient of 7 to 8 national and international awards for academic excellence, innovation, and contributions to pharmaceutical sciences. He is peer reviewer of various international journals and publishers. Recently published a book on multifunctional nanocarriers, biopharmaceuticals, and nanotechnology. Research and development of complex, innovative, ophthalmic, injectable products, including micro and nanotechnologies for regulated quantitative text for various government and institutional bodies, quantitative science and technology.
Quantitative test for
He is also
glucose by
for the various international journals.
Glucose by
he is the editorial board of the
Benedict's method.
journal being published by the Nature Sciences. According to the announcement
Glucose by Benedict's method.
Based on the database and basis of standardized citation indicators, Dr. Mehra name is included in the top 2% scientists in the world in the area of pharmacology and pharmacy, consecutive for 3 years. His contribution continues to influence academia, industry, and healthcare innovation in India and abroad. With such a distinguished background and wealth of experience in the pharmaceutical domain, we are truly privileged to have Dr. Nilesh Kumar Mehra with us today. We are confident that this session will offer valuable insights and inspire meaningful discussions. Without further delay, I now invite Dr. Nilesh Kumar Mehra to kindly take over the session and share his expertise with us. Please go ahead, sir.
Thank you. Am I audible?
Yeah.
Yes, sir. Audible, sir.
Yeah, thank you for confirmation. So I will share the screen and I think this slide has been visible for you all and I am also audible for. So good afternoon, everyone. So and I would like to thanks for the entire team who has been organizing this faculty development program, so that the new things and the novel approaches what is going on in the pharmaceutical science, including biomedical science, so that could be somehow it could be beneficial for the all faculties who has been attending and joining these sessions regularly every day. So I congratulate to entire team of this KBP since Vijayawada for organizing of such kind of this wonderful events. So today I will be discussing about some things different and hope the whatever I'm informing, sharing the different domain of this technology which is going on. So that's a definitely it could be helpful for all who is working on this particular segment. And so the title which I has chosen today, the innovative drug delivery approaches in combating of the triple negative breast cancer. So definitely we know about the today's what happened in the therapeutic approaches and the different kind of the approaches for development of the pharmaceutical new product. So that definitely first we will previously we are working on a different segments including of the different particles, like starting by the particles, then now we are working toward to the nano technology or nanoparticles. Everyone know about that, the terminology nano particles, and then we know about this the size of we talk about. So working very small, thinking about to be toward to the big. So that's a nanoparticle or nano science has to be taken up. So the nano medicines we know about that the size of the nanomaterials. Now no, the different kinds of the materials their size is fall in the under nanometric science. That could be helpful only like not only in the pharmaceutical science, in other field also. So that could be helpful for in vitro, in vivo, as well as the clinical output of this one. And different types of the terminology we are using. If we are talking about the nano-oncology, nano-pharmacology, nanotoxicology, so the all segments, initially we talk about the in a different segments, diagnostics, and imaging agents, analyticals, or the different also in different segment. So all the things are related with to the nano technology. So we know about that. Everyone has the faculty point of view, but I am just giving the brief about toward that and try to connect with how does the different kind of the dosage form or the pharmaceutical product has been interlinked with us, and how we will be try to improve their problems or the challenges they are having. So the most of the drug deliveries that we had developed, we definitely initially we talked about the topical things, then we had talked about the oral type of the dosage form, and definitely like a parental routes also for the parental drug product. But that is depending upon to the invasive techniques as well as non-invasive point of view, because it's the product has to be more patient compliance and patient convenience. So those problems which initially we face it, and like this potential due to the and degradation of the APIs, once they will become come out from the dosage form, they will become in the biological mill, and the so the what happened in that case, the cost of the if this dosage form has been increased, and we are trying to do the more giving the more doses to the patient, so the patient compliance will also lead it. So that's why the increased frequency of dosing, faster degradation of the APIs and all, so the toxicity will also leading. So that's why for to solve up those kind of problems, those are the common problems which have been firstly found in the oral like a tablet or the capsule dosage form also. Like so later on, that is not a new one. I can say it's like long back. I believe it is 1905, the 1900 years, more than 100 years ago. The people as Paul Ellis, he has thought on that particularly different kind of the concept, that is drug targeting concept. Yeah, is thought of about this German scientist. The concept for targeted drug delivery may not be new, but cannot be we can be suggest that with the help of what we have been, by changing of the some things onto the particles, they will be fall in the micron as well as the nanoparticles. So do use of this nanoparticle or microparticles, or we can be alter of those the surface of that one, so they could be work as a drug targeting approach. So if we go in a way of our Indian perspective of point, we know about the how the targeting will be happen in during this why the concept of Arjuna. So that's a magic bullet for effective drug relief. The people has been work on that one, and somehow we got the success in some product is available in the market also on the targeted drug delivery waste system. So this targeted drug is nothing is like this one. We have to be target and we have to be designed that the medicament has to be go in a right place in the body at the right time with the right dose with the right time period. So that's the major things we have to be do, because it's a huge body, human bodies, and whenever we are giving the oral doses from oral route with the oral or any other, like not only oral, but in case of a parenteral of some injectable product, so that though what they will be traveling in inside our body, and all other cells they will be impacting on. So what happened in that case, because that somehow the drug has been traveling in that, when we know about the pharmacokinetic point of view and the biopharmaceutical aspect, so the drug is unnecessarily going in some other region also. So that's the one problem we are having, because in that case, like highly potent drugs, if you talk about a cancer therapy, the highly chemotherapy, they will be traveling the molecules in the other portion of body, and they are creating so that different side effects. So yeah, the patients is battling against with the like cancer, and another potent molecules they are going in a normal, they are also not supporting. So that's why this targeted drug delivery concept has been came in the picture, and we have a different kinds of the new generation nanomaterials with us. So the first thing which I want to be why work with the nanoscale regulatory system, the definitely to minimizing side effects, and if we are able to be reduce the side effects of the API, which are highly potent in that one, and definitely we will be able to be maximize their therapeutic effect. So that's with the help of this targetable drug delivery, new not only the targetable, present a target site in the desirable optimized sufficient quantity of the concentration, and depending upon to the type of the size of the particles, they will be go in a body, and then different, like we know about the first order, second order, or the central compartment as well as the peripheral compartment. Like if we had gone I had collected the information like a small particle, like those having the less than of 10 nanometer, they are lost due to extravasation and mainly absorbs into the tissue. Large particle like more than greater than of this 200 nanometer, they quickly capture, and they have been go excreted out. Particles between 110 and 70 nano, they can be kept more penetrable with the help of capillary vessels. Particle between 70 and 200 nano, so longer circulation time. So that's kind of the things we have been gone with the, to understand about the different size of the nanoparticle, how does they behave in the body in a different organs, cells, and for a pharmacokinetic point of view. So that's nanoparticle we have been easily manufactured, we easily fabricated out. But the thing is like that, not only the easy manufacturing or fabrication of the nanoparticle or microparticle is not enough for us. So we had work on that, but we did not. We have want to be, some kind of more again, some modification on those nanoparticles, and yeah, it's not also easy to modify those one, and after that to control it. So that, but we have to do some kind of the chemical grafting or functionalization that we call it, to provide something with a different functional groups with a chemical functional group, and by attaching on the particular API or compounds, so that they could be act, or they can be helpful for, to go by this targeted village. So that's also not a new one, but surface modification is kind of the modifying the surface of the materials is not only the nanoparticles, some APIs or the some things that we can be change their by properties of physicochemical as well as the biological characters. So that's the one things with the help of this functional. So that's what happened, because initially I talked about that the when conventional they could be the API will be go firstly release, and there will be chances for degradation or conversion in another form also. So definitely if we go on the concept of like this, we know if from like that the first things, because the drug is very important for us, and that has to be circulated in body and reaches into the different organ cells or wherever. But the thing is like that they are not able to be circulate a longer period of time. They can easily excrete out, and either they can be degraded out. So if we talk the concept of this Eskimos, they will be, you can see the picture of this one on left side of this one and right side also. This is a long back. If you are taking this concept for from our daily life, this one, and you implement it on this nanoparticle, so that's the try with the help of like Paul Ellis. We have what he has thought, and we had to done along with this concept. So these are like for example of this nanoparticles, why which this is the simplest one. Different kind of generation of the nanoparticle we have it. We know about that one in a theoretical as well as in the experimental. But if we talk about the I, because I had worked in some industries also, to we being a faculty or being some academia, we are easy to based on this concept we can, oh yeah, this we can nanoparticle we can be prepared, we can attach on the PEG chain on the surface. Yeah, definitely the lipids has been also prepared, or like any targeting ligands we can attach it. But definitely there this is not easy to handle on and to control of the properties of the ligands or the pegylation or this one, because in simple kind of the formulation where the multi-unit operation has been involved, they are not easy to control of the other parameter also. So like we call like a quality target product profile, critical quality attributes of the dosage form. So that's the variation in those CQs and this quality target product profile happening. So those are controlling the controlling of each parameter during to the manufacturing in process, and even though after the completion of the finished post drug product development. So that's why I'll give you the one glimpse on this particular complex product, as this I had taken from with the US FDA. So those they may be closely work in the product development. They may we go more deeper on that one. So previously, what the we are people we are working on a simple product that we call like a non-complex or simple product. What happened like that? You can be see like one artificial this lubricant eye drops. What happened in this? Like this is one watch. They are having the like a hydroxypropyl HPMC one and the preservative and some kind of maintaining of the pH and osmolality. So that is easy to be formulation point of view, because there is no hurdle in the API's control of API physical properties. The multi-unit operation has been not also involved, and this other and those. Well, we talk about the complex product. You can be see like this is also available in based on to the liposomal product doxin, where the control of the doxorubicin or hydrocolloid, which is also their parameter is quite tough to control when we are developing one product. So the API's characteristic has to be preserved, because ultimately they will be go release out from this product and reach into the biological milieu of human body. Sorry. Another word like Respiron, Consta, this is also based on to the long PLGA microsphere. This azopt, that is a combination of brinzolamide, ophthalmic suspension, because in that the brinzolamide having some different challenges. They are like a sterile and the non-sterile one, because the product is fall in the BC class 2 and having the poor aqueous solubility. That's why we are able only to do develop up to the suspension. But the suspension is not only that, we can be like a particulate suspension has been suspended one, but after that, because the brinzolamide having the high particle size, now we have to be convert to the lower particle size like that. Another product like Durozol, this is also one emulsion product. In the ophthalmic segment, these are like a newly I think only launched in the 2010 or 15 some before, and one product has been also launched in 2018, that is a combination of brinzolamide and bromelain also like that. So those suspension, because these are coming in the complexity toward to the APIs, toward to the dosage form, toward to the route of administration. So that's I have taken from USFDA one, and this is also I have taken from the USFDA that guidance from the industry. The complex product is associated with the complex API like a peptides, natural synthetic polymers, peptides, and the high molecular weight drug compounds also, or those having a highly potent physiochemical properties of the APIs. Complex formulation like a liposomal, colloidal formulation, suspension, emulsion like that. This is also not because their multi-unit operation has been involved during to the fabrication of them. There. So dosage form like intravitreal in case of ophthalmic, you can see like the ophthalmic medicament or the eye drop, they are not able to be do like permeate the different barrier which are there in the eye, and they will not reaching to the this vitreous region of the eye. So that's the one challenge. That's why the people has developed of a intravitreal injection. That is a invasive techniques. But what we wanted, the vitreous posterior segment of the eye related as disease has to be cured and treated well. So that's a one problem, like a complex route of delivery, like this in case suspension topical. And another one segment has been recently introduced, that is the concept of a complex drug device combination. So that's the new era in a case of the medical device section also. And we know about that some already product has been in a market. Those are based on the drug device combination like auto injector one, and this is an intravitreal injection to deliver the drug on a posterior segment of the eye along with the contact lens also. So those are like that's a pharmaceutical equivalence is major challenge to achieve for complex API. Not only in the innovator point of view, I can say in a complex generic formulation is also very tough to develop along with the bioequivalence also. So those are about the changes in the from this simple product to the complex one. Because the simple product the market has been already gone, and we have to be shift toward to the complexity, and we are been to be familiar with the new technology or new machineries. Those they are involved to develop of these complex drug product development. So evolution of drug delivery system. This is also one very beautiful picture and summarize, which has been published in Journal of Controlled Release. I have taken from that one, and this is a complete picture, a holistic picture of when we long back, like we had initially thought about the dissolution and diffusion control, kind of the dosage form only. Then later on, osmosis based principle for OROS, the technology developed, and ion exchange. Then later on, like this, this Lupron depot PLG nano microparticle. So initially, based on this system, we have been introduced into the micro technology, and initially we had developed one PLG microparticle as a depot system. Later on, we have been shift from the micro technology toward to the nanotechnology, nanomedicine point of view, and we have like this product, like you can see in Taxol, that is a paclitaxel bound in a pegylated castor oil. That Abraxane is the paclitaxel bound and albumin complex, human serum albumin nanoparticles. So those are like a doxil also, that is a pegylated liposome, that is a nanomedicine also. So we are working in this particular segment from like I can say after 2000. So based on this successful full of story of these products which are already available in the market, definitely whatever the new things we are working throughout this one, those other products will be come in a market later on also. So that's the picture from long to summarizing of the successful story of the all from previous system, previous mechanism, and micro as well as a nano technology or nano particulate waste drug delivery system. So this is the one picture you can be see the liposomal formulation where I can be share my thought process on that how we will be to get the information on those products, because you maybe know, some maybe know, some may not be know that how we will get the information from the already available market. So maybe the along with this doxil products or everything in this one, you will be get the one label, that is the one leaflet is always with that, and there is some certain kind of the guidance for that, and we have to be the industry or innovator has to be follow on that, and those information are very crucial. You can get these information like a in terms if those they would want to be developed of this generic version of this product, they will be get the qualitative information, but the quantitative information is not there. You have to be go on the reverse engineering, and then you will be find out the quantitative value of the this doxil are there in there. So that we know about the composition like that. These are information like this API doxorubicin hydrochloride salt form is there. MPPEG DSP is also there with this particular formula, and HSPC also. After that, cholesterol also there, and some information we got from the other papers like that. These are the how does the electron microscopic of this doxorubicin is there. You can see on that one. Those are later on the people has developed. They were buy it of this innovator product, and they had gone on to the this reverse engineering to get the information. So these are all complete picture of morphology of other product like a onivirine and depoform. How does they would like on the surface morphology. Another I will be give the example of PLJ microsphere that is based on the long-acting injectable. The brand name is available on a Vivitrol. Vivitrol is the information you can get the composition point of view, and this is the one information how does this naltrexone. This is the API, and Vivitrol is a naltrexone. It is work on the opioid kind of things for psychosis patients. So that the people what the motto of to develop of this PLG microchip. We know about the PLG polymer. They having the property of that they can be release the pattern this naltrexone up to like a 2 month or 3 month. So the patients or those are psychosis one, then it is not easy to be injected on daily basis or like a BID or TID. So that's why the people has thought and they had developed this controllable kind of the release rate, and it could be go up to the 2 months or 3 months. So that's once we inject it, they will form of a depot, and that will be released continuously with a sustained and optimized ratio up to the 2 months or 3 months. So the problem is this is also one of example of complex product because the naltrexone is also we have it to control the properties of the naltrexone, and then the another like a PLG, that's a one challenging polymer also because you can be see the PLG is also the CPP and the critical quality attributes of the PLG selection is also very crucial and important. So that's we have to be select out because the PLG is available in a different form, rhizomer form, and based on the lactic and glycolic acid, they are available in a 50-50 ratio, 65, 35, 75, 25 ratio. So we will be check out that how their property or the different types of the polymers, this PLG will be helpful to develop of this product. So that's the one problem to control of that. Another like homogenization is important to control of the speed, time, process, temperature, and these are all also to check out on the particles and the particles size distribution, their drug distribution in different phase, encapsulation efficiency. Those are impacting on that. So what happened? This is easy to do on a bench level or a lab batch, but when we are transferring this technology to the manufacturing plant, so what happened? We have to be concrete written document on a batch manufacturing code, and then the whoever is working on a like operator on any technique worker, they will be follow those or the scientists. They are follow the whatever the written document is there. They could not be deviate because more than like this. For example, let us talk about the this homogenization. The speed is supposed 20,000 is written. So that's the one problem because there is not they had not provided a range. So always like a 20,000 to 22,000 or like that. So that we have to be check it out because they will not go beyond or not going like this a lower one, highest one of that. So that's some problem or challenge we will face into the manufacturing plant when we are scaling up the batches of this kind of the complex product development. So this is also one very important. The people has work on that. Like you can see the how the degradation behavior of Vivitrol during to the in vitro release. Yeah, that because we had taken off the 75 to 25 PLG, but you can see it on that one. This information we had got it. Each dosage strength of 380 mg naltrexone per vial is there. Naltrexone is incorporated in 75 to 25 PLG ratio at that 337 mg of naltrexone per gram of microsphere. So that's the one other composition like CMC polysorbate 20, sodium chloride, and those information we can get. If we want to develop of a complex generic on that one, so definitely this Q1 formula is here. Now we have to find out the what the Q2 value are there. Then we will be take the approval from the USFDA for even the development of generic form. For this one, what I want to be tell, the people have thought, how does this PLA microsphere behave when we are subjecting for a long term? Like a 1 month, 35 days, they had done the study. So they had found initially the first day, you can see that there is no change in the structure of the microsphere. Third day, fourth day, seventh day, not like that. But like this 7 day, 10 days, and 14 days, there is some kind of the degradation onto the this initial mid of phase. Like I can say, like initially within 3 weeks, they have been found out some kind of degradation on the PLA micro. So they found out that the later on after that they will not be the other particle will not be degrading. So this is kind of investigation they had the people had done onto the even though already this developed product also. So there is a new kind of the investigation also that, and definitely while submission of the uh, data of for getting approval of Vivitrol, they had also find out or the FDA may ask some kind of the question also those days. So the release we know about the either they will be work on to the self diffusion release from the surface or erosion of the polymers or like that. Those mechanism we know about the other diffusion, dissolution and all. But what I am believing in the concept of the this complex product development, we have to face the major problem in case of the characterization. The drug product, if we are going in a more well-defined characterization of all the evaluation parameter, then definitely the product will be going a like a successful in getting the regulatory approval. So these are the all listed like in case of the nanoparticle like surface charge, particle size, shape, PDI, and surface. All we have to be do carefully, and that could be repetitively one batch to batch variation. We have to check a man to man variation, person to person, or the instrument to instrument. Those are all we have. If we had checked initially, then definitely these characterization parameter will not be changed, and we will get the proper drug products for, and we are able to be submit for the approval also. So this is I also already had given up. The one another I have taken the ophthalmic nanomedicine because here the more important thing is that they had shown even though only the component Q1 value is there, but the Q2 value is not given. But you can see there is a line on that one. So this is a hidden document, but the percentage bit by volume is there, but the form of or the APA, the sodium mediated tetrahydrate dihydrate or there or like the quantity has not. So after that also the people has filed the Q1 Q2 value formula, but some problem has come and they did not get even though the Q1 Q2 approval also. So those are like a challenge is still in the complex generic formulation also. So definitely for development of the any products, we have to follow the using this inactive IID database. It is available on a website, inactive ingredient database. So by choosing of the proper this excipient, we can be choose the proper dosage form, their route, and the maximum potential of this drug or the excipient we can utilize in the development of the products there on based on the safety and toxicity data. Beyond that, we could not be used, and if we are using that, we should be giving the justifiable all the data in vitro, in vivo, and the preclinical also that. So that could be helpful for development of the product also. In the development of this product, the QVD approach is also there because I had worked on the closely on this. What does impact on the QVD into the product development? It's like we can say as per the QVD with ICH Q8 also that the systematic, holistic, and proactive approach to manage the pharmaceutical product life cycle with the defined, predefined objectives, with the controlled increase of the process and product to understand about the API, to understand about the excipient, to understand about the API excipient compatibility, interaction, to understand about the process and how we will be control onto the process, so that we can we say that it could be the product has been developed and there is a less risk onto the quality, so that QRM has to be minimized also. So that's we know about that, because the people previously working on now NDA as well as ANDA, they were thought about that once we develop the product, then we will be learned later on, or we will be develop the product, then we learn it and file the product. So the whatever the what happened in that case, there is chance for a batch failure, stability failure, even though the regulator will ask this amendment. So this what happened in that, there is a what we will face the companies or the those the people, they face the approval later on that one, or the market product has been reached in that the post compliance has been come like that. So that's for that QBD will be helpful in that way, because this could be mitigating the risk initially, and we will work holistically, so that the later on we will not face like those problems. So it could be helpful for higher level of assurance of the product quality. The regulatory compliance will be minimized, and definitely minimizing eliminating of the potential compliance in the or increasing efficiency of manufacturing process, and the less chances for the batch failure, or less chances for the formulation revision, as well as for the component also. So now coming to the breast cancer, I had particularly work on to that coming currently on the triple negative breast cancer. So the TNBC we know about this kind of a breast cancer that has does not have any of the receptor that are commonly found in the breast cancer. So there is a distinguish between among the breast cancer as well as for the TNBC. So initially what the people, they have been like a positive of this TNBC, but they are not able to be diagnostic of this TNBC at early stages. So that's the one problem. So if you think about this CDC from this one, think of cancer cells as a house and have three kinds of locks called receptor. So one is for estrogen, progesterone, and HER, human epidermal growth factor 2. So this is a one house. If you think that if you cancer test positive for these three locks, which are known as a receptor, estrogen, progesterone, HER2, then doctor have a few cues to find out that how does go and then kill to them and destroy it. But if you have pre-NBC, those locks aren't there. So the keys doctor usually use won't work, because we don't know what the kind of the medicament has to be prescribing on that. So that's the one problem with this triple negative breast cancer is known about that negative one, estrogen, progesterone, and HER2. So we will not find out any lock keys to lock open this door, so we can be go inside this house, and this the APIs will be go inside and they will be kill it. So that's the one problem. So 15% of the breast cancer is almost triple negative. So that's the one problem while diagnosing or of this TNBC. So epidemiology, there is a huge number of the patients, they are suffering by this breast cancer as well as for the TNBC particularly. Another, these are the some characteristic clinical feature for this breast cancer as well as for the triple negative breast cancers. And these are the thing, some stages. Initially, the people, if we are those people, the patient has been a TNBC, they are not able to be detected zero or early stages. So that's the one problem is there. Later on, they are usually come in the picture, and the when metastatic kind of the cancerous cells are there. So that's the one problem in case of the diagnosis as well. The diagnostic another technique also like we are usually using, but they are we are not able to be detect. So previously the people at early stage, and later on is developed, so that would not be even though treatable. Then surgery is the one option for that. That is recommended for as per the this stage 1 and 2 and 3 4 like that one. And chemotherapy initially we are giving one, and these are the marketed nano formulation Doxil, Abraxane, and Zenoxil coming in the picture to treat of this kind of the that one. So I'm giving a one novel things which maybe you not be aware about that one. And these are the which we talk about the drug device combination in case of medical device sector. So the single entity as per the USFD, this one you can see single entity combination product comprise of two or more regulated components, but it will be co-packed in the combination products. It will be cross-labeled combination product. So this is a product generally based on the primary functionality. So this is one thing, and you may, this is the one example I have taken. We had work on this particularly drug device combination along with the medical device department, and because this is the one product available in an innovator point of view, the cost of this particular marker clip is 7,000 in Indian rupees, and it is available in as a marker clip. So what happened? What is the useful of this marker clip? This is having the, some kind of gel on side that. So after this post surgery of the breast cancer, the surgeon has been implanted of this gel onto the nearby that region, and they will be observing of the development of the reoccurrence or the how the this cancer cells are going to be destroyed or like minimizing that one. So that could be helpful. And but this is a single use one, and the cost is 7,000 so what we had worked with the closely with these surgeons, and they suggested that why you will we not develop some kind of the this one, and we had working on that, and even though we had developed that one product, and we filed one patent on that technology, and one before that our development. This is the one other tissue marker plex people have been worked on that is kind of a biodegradable breast tissue marker clips has been already published in another thing. They have been taken out the PCL, PLJ kind of biodegradable polymers, and they develop of this one. They injected on that particular region, and they have taken up the in vivo X-ray imaging, ultrasound, MRI also, and they have been how closely that could be this kind of the marker clip will be helpful to diagnosing as well as imaging of that way. So that the we have to be thought somehow. How does this achieve this triple negative one? So that could be easily diagnosed as early stages. That is the one major innovation in this field also. So that's the one things we have to be like that. So there's always the innovation and the new things always welcome on that. And definitely we have been work on that some kind of optimal ligands for drug targeting, so that this TNVC has to be minimized or mitigated. The different kinds of the ligand driven drug targeting has been explored in this breast cancer as well as TNVCs for by active as well as passive drug targeting approach. They could be wind up, and with the help of like a and the technology, like we know about the receptor mediated endocytosis. These two scientists they had explored in 1972 of this the pathway of RME. We know about the endocytosis, and this is a new modified version of endocytosis. In that one, the role of the type of ligands, type of receptor, formation of elatrine coated vesicles or cabule one, and how does it will be convert in the early endosomes, late endosomes, lysosome, and it could be releasing of the pattern of that. So that we know the RME occurs when receptors on the cell surface are bound by their ligands, and it could be internalized with the help of this clathrin as well as the cabule coated pits. This Clathrin and cabule are nothing; they are just like a membrane associated protein pits and play a major role in formation of this one. And that could be helpful once they form up that become endocytosis vesicles that we know about that. But what happened in that there is a change in the pH altering of that one. The early endosomes having the slightly acidic pH 6 to 6.5, that become late endosome once is that the pH has been changed from 6 to 6.5 to 5 to 6 that one. So that's the one things we have to be do, and it's not only that the product has been the drug has to be reached onto the surface of the cells. The after that reaching of that one, how does they behaving in intracellular or find out the region. So these are also different kinds of the receptor and the ligands are there. We know about that, and everybody is working on those one to find out some more important kind of the results or solution. So we are able to that. So this is a picture. You can see the early endosomes how it will be converting into the early endosome about to undergo the conversion, and after that these endosome will be go and recycling again. So and same way like a receptors here also they can be go, and later on it has to be work on the and that depending upon the type of the API they could be work and to this intracellular. So RME definitely type of ligands we had work, type of receptor, elatrine coated pits, early endosomes, their pH change, late endosome, their change in the pH because the hypoxic condition also there, acidic kind of this there, and later on this recycling of endosomes is there. The people has already, like, it's not only the theoretically point of view we can be quoted, but the people they have been worked closely onto that. How does this RME have been take place? So with the help of electron microscopy, with some what we have do in that case, such along with the cell lines on this, while we are doing the experiment, we have to be connected with the electron microscopy, so we can be observe each moment, each step of this kind of the RME approach. So that people has taken the beautiful image. You can see they are labeled by the E, that is a E label for EG, early endosomes in case of EGFR, and late L is indicated for lysosome that are visible. M is for multivesicular this one endosomes that we call like a late endosomes also that. So that's the one we have been to work on that one. Now I am giving one little bit info one projects which I have we had done on latest on the one particularly palbocicillin that we know about the CDK4 by 6, that is cycle independent kinase inhibitor. It was approved in 2015, and so far only the one product is available in the market, which is evidence, that is a palbocicillin capsule only in a 75 mg strength. So that's the one we have designed, this targeted, and it was published in the IJP. This is the entire team from the by including of other biological peoples also. So that we had developed polymorphically loaded lipid polymer hydrogen nanoparticle with this nanoprecipitation method, and that is we had definitely the optimization of the all each parameters of the process control of the PLG selection, PLG type of selection, and molecular weight kind of those things, and those initially we had developed based on to the quality by design and based on to the this optimization techniques also. So that would be helpful because it will be save our time. It will not be lead to the wedge failure. It will not be taking that so much time to what I can say, like previously we are working on OFIT technology, that one factor at a time we can be optimized, because we are working on the complex product. So the product each sequence of API or the sequence of the exhibit we have to be take care, and later on what the techniques we are using their CPP critical process parameter and all these things we have been to be take care to develop that some of the product also. So that now coming to the characterization, that we had done the exhaustive characterization on that, and we had before this characterization we had done the optimization with the help of DOE also. So these are the why like different FTI and XRD and DSC NMR. We had done the exhaustive characterization, and the team has worked on that one. And after that we had find the optimization of this particle size and PDI, that is well in range, and we had done the repetitively. We have performed like experimental to expand day to day change and man to man change, machine to machine change also, that so that the product has been like in a developed in a robust way, so anybody can be work on that, and hopefully that will be more reproducible and all this thing. So the beautiful lecture of this lipid polymer hydrogen nanoparticle with the help of TAM images you can see here on the right side also. And after that we have subjected for the release pattern up to this 72 hours, and this is the closed structure of the up to 2 hours initially, that how this it will be releasing of the drug pulviciously from the targeted and the at a different pH, that is the 5.5 as well as 7.4. 5.5 we have been selected sodium acetate vapor because the pH of the life this cancer cells is quite of an acidic one. So that's a release of a controlled one and sustained one. And we had also checked out that how does it will be the kinetics is one, and which mechanism they are following to release of the release. For that also we had worked on this different the analytical resolution techniques which are available in the market also, that, and we had find out the dialysis diffusion they are giving the better results on that one from this release pattern. Definitely, as per the ICH, we have to follow of this to develop of the product when we subject it for stability study. Yeah, though still we have a problem that the stability or the some guidance are not there on the nanomedicine. but the USFD and other regulatory agency, they are working on that to develop up that one. Like some, even though not in vitro release guidance is also not there. So those they are distributing in different universities or research, and they will be collecting data and they will be time to time publishing on that. So here we had selected two different temperature stability condition. One is a 2 to 8 and another is 25 room temperature, because this is we generally recommended a 2 to 8 temperature. So that could be work as a accelerated condition, and the data in terms of particle size, PDI, GTP and entrainment is quite well up to 60th day. That means the 2 months. So that's a quite stable product into that one. So after that successful in the stability, we had gone on to the in vitro cell line studies. We had selected the three different cell lines. Like we know about the MCF7 and MDMB231. Those are known for the TNBC cells, and we had initially subjected for and check out the how does they are in working on, particularly when we treated on these cultured cells, and we found that our targeted this folic acid polylipid hybrid nanoparticle is quite giving the better response as compared to the plant drug also like that on the MCF7 at 24 hours, 48 hours, and well 72 hours. And we had got off this IC50 consultation of plant drugs as well as this one, and we had compared with this how does it will be change and how it will be improved on that one or superior with the plant drug also that. So we have found out in like not MDMB231 is the TNBC one that 8.26 at a 2 or 24 10. So almost like a, we can say that 10% is increased, 10 folds has been increased onto the efficacy of the this with the help of this lipid polymer, lipid hybrid nanoparticles. So that's giving the where more success toward to that. We had done on the quantitative cellular uptake also that how does with the help of blocked as well as receptor block on this both MCF7 as well as MDMB231 cells, and we found better cellular uptake from this FA or targeted drug delivery system. That could be indicating that yeah, definitely we have to work on that, that it is better things, and only the we have to be develop the robust formulation uh, of this lipid polymer hybrid nanoparticle. This is the also qualitative cellular uptake. With the help of that, how does they will be realistically going on to the inside and how they are so more fluorescence of this indicating in MCF7 as indicating in MDMB231 at three different point 2 hours, 4 hours, and 6 hours. It is giving the quite well like a uptake of the formulation. So that's one also other assays we had done on that. Another important assay which we had done on this particularly on MCF7 cells as well as on MDMB231 cells. We had done this is like a migration assay, and we have to on this one we can calculate the distance how this is in case of control, it is in case of planned drugs, it is in case of targeted. So that you can we see how does at a 24-hour different time points that they are the migration toward to the this dead cells or this cancerous cells is minimizing. So that's the one that we had calculated out the percent cell migration inhibition on both cells MCF7 MDF, and we had statistically compared with that, and then we found a better significant results on that one. This is also we had done on case of apoptosis assay, in case of MCF7 cells, in case of MDMB23 cells, and we found out the higher intensity of fluorescence uptake toward to the late apoptotic assays. Later on, we had developed one spheroid culture that is very important nowadays. It is like a subsequent or substitute for the in vivo study. We do not be directly jump onto that one, and we have to be develop of some kind of spheroid. This is kind of a bunch of group of this cancerous cells. You can be see these are the realistic picture from our side, and with the help of microscopic techniques, so that we have to similarly in vitro cytotoxic assay on this 3D the different little bit the change of different kind of the ball plates and other things has been required to develop of this spheroid, and we can be check of this mass or the volume of this spheroid, and we can compare how this mass, the volume or respiratory volume has been changed if we treat it with the palbocicillin as well as the targeted formulation in that one. So that is giving to the better response in vitro tumor volume also. So that's the picture. You can see like day 1, day 2, day 3, day 4. In control, it is accelerating one in increasing, but in case of that one, the growth of these groups has been decreasing. But even though not only in case of a day 4 only. So that's the one. Later, based on this success of the story, we had developed this Volvo based on the pharmacokinetics, and we got the better pharmacokinetic parameter in terms of the clearance mean residence time. It is been increase of that one, so that it will say that the better plasma drug concentration profile we got after IV injection at a dose of 10 mg per kg body weight. The toxicity is also giving us a better response, so that we can say that our product is not giving any toxic response, and this product is we somehow giving the more higher anti-cancer efficacy as compared to the palbocicillin alone. And if we can compare with the tablet, that's we will got that better things from the dosage form. So that's the one we had continuously working on to that one, and this is another paper we had published on same drug, but we have chosen the another chitosan coated carboxyphenyl uranic acid conjugated chitosan nanoparticle for that, and aligned with we had developed these same things and same approach till to that one, and so that we got also those and published in an international journal of biological macrobiology. So preclinical evaluation, definitely it is also required, but we have a to initially if we had developed of the product or robust product, then definitely we have to initially look on the in vitro and in vivo the cell lines, or even though with the help of respiratory model also like. So that's the one we things has to be go, then not directly jump onto the animal models, and that if we got it the better response, then we have to be go on a preclinical evaluation also there. So that's a definitely filter validation is also important in the injectable. Like this is a parental product, we have to be select. We have to find out the to either what kind of the filtration has techniques has been utilized. So that could be helpful for sterilization. So from my side, I can say that the future perspective is that if we are having the multi-dimensional people in the team, if we have a collaborative people on the different institutional multi-institutional, multi-university, or like that, then definitely we are able to be develop of some the better complex product. Also that the complex formulation, and later on it could be translatable, and it could be commercialized, so that the people will be get the benefits also. These are some glimpses which I have been from my another like this one some prototype product we had shown on different platform, and students from my labs, and this is recently edited works from my side also being an editor that could be helpful for your institution or universities. And this is my phone number and email id. You can be connect with me for so that I can be help you in any way for practical as well as for the uh, like that we can be helpful. We are we could be helpful to develop of the products or any not only in a product. I can be helpful in research and academic and wherever I can do some kind of contribution that. So that's thanks a lot from my side, and the entire team also. I would like to congratulate on, and you had given me opportunity so that I had said something which I can during this during to the working onto the research, during to working while discussing and working among with the students, among with the different PIs, and then every day we are learning something. So that's a good. Thank you for all. I would be happy to answer if somebody having some doubts or the queries. Right now, if you don't have, you can connect with me on the phone as well as on the email ID also. Thank you.
Thank you very much, sir. Thank you very much for your valuable presentation. And so that is the enlightened speech. And you have highlighted about nanoscience, drug targeting concepts, and nanoscale drug delivery systems, and so many things about nanoparticles, simple versus complex products, evolution of drug delivery systems, long-acting PLGA microspheres, complex ophthalmic nanoemulsions, quality by design, role in product development, biodegradable test tissue marker clips, receptor-mediated endocytes. So preparation of lipid polymer hybrid nanoparticles, stability studies, and so many things. And so we got some idea about it. And actually, there's a problem in audio, so we're unable to hear you very clearly.
Oh,
Yeah, but some disturbance in audio is there. Even I cannot hear you clearly. So by seeing your presentation, I got some knowledge, and a very nice presentation, sir. Thank you very much. So thanks for your presentation, sir.
okay, okay, no problem. If you, whoever is having some question, query, they can connect with me.
So for any doubts, we can contact you, sir. Just for
Sure,
that. And
you're
so
most welcome.
very nice to see you, sir. Thank you very much.
Thank you.
Both to Dr. Aripa.
Thank you so much, sir, for sparing your valuable time. On behalf of organizing committee, we are very grateful to you. There's a small announcement for the registered participants regarding tomorrow's session. Dear participants, tomorrow's session will be at 3 o'clock. Tomorrow afternoon, 3 o'clock. The session will be by Anand Kumar Chettipalli, sir, from Galgodias University. Dr. Anand Kumar Chettipalli, sir, from Galgodia University. Thank you so much. We'll be sharing the attendance sheet for today's sessions in the WhatsApp group. Please fill it and submit by today itself. Thank you. We are concluding the session. Thank you so much, sir.
So this is the end.